So Dr. Witowski forged his early career in natural products chemistry. He received his PhD from the University of South Florida while researching bioactive compounds from marine invertebrates and microorganisms. This foundation allowed him to transition into the burgeoning cannabis industry, becoming the chief scientific officer for AltMed, a multi-state operated cannabis company. While at AltMed, he pioneered new delivery systems of cannabinoids, including transdermal gels, patches, and metered dose inhalers, while overseeing manufacturing of the Movie brand. To date, product sales have exceeded $100 million, and AltMed has merged with Verano Holdings in a multi-billion-dollar go-public transaction. Dr. Witowski now leads Psilera Biosciences, a psychedelic-based biotechnology company developing its proprietary neuromodulator pipeline to target cognitive, mood, and substance use disorders. Is that Psilera, or how do you pronounce that?
Psilera.
Psilera will usher a novel DMT delivery device into clinical trials while new psychedelic analogs are undergoing lead development for IND submission. Thank you.
Thank you for the introduction. I will say I'm getting a little bit of feedback in my headset. I had to use my headphones just because I feel like my hard drive's about to take off on me. It wouldn't be a virtual conference without a little technical difficulties. But how's the audio quality?
It sounds good on my end.
Okay, great. Let me go ahead and share my screen. I assume you can all see it.
Yes.
Great. Well, thank you for joining me today. I'd like to talk about my company, Psilera Bioscience, and the next generation psychedelics for mental health disorders. Let me get into presentation mode. So I'll first go through an overview of natural products, and this is very near and dear to me as a natural products chemist, and how these compounds really serve as a foundation for drug discovery. We'll overview some of the bioactive substances found from natural sources and look at some of the areas of indications that these compounds could be useful, including mental health. We'll then transition into psychedelic as an overview and how these can be useful for neurology as well as psychiatric disorders, and look at the indications and market potential of some of these compounds. Lastly, we'll do an overview of Psilera and look at our preclinical pipeline into our clinical developments of psychedelic analogs, as well as new delivery devices. So every few years, the godfathers of natural products chemistry put out a review article where they go through all the new FDA-approved drugs, the various indications in which these compounds are have been approved, as well as whether these compounds are naturally derived, are they purely natural compounds, semi-synthetic, fully synthetic. So since 1981, 65% of all FDA-approved drugs are either natural products or derived from natural products. So obviously, this serves as a very good foundation for the development of FDA-approved drugs. And these compounds are found kind of ubiquitously through nature, whether it be foods, plants, mushrooms, other nutraceutical products. And these compounds really are complex, and they're also included in a very complex matrix, such as the extract or the cellular environment in which they're produced. So some of these inherent properties of natural products make them useful, but as well, looking at how they're produced, they're generally secondary metabolites. And these are compounds that are not produced for primary reasons, whether it be for reproduction or for cellular purposes, but they're produced mainly for defensive purposes, whether that be against predation, feeding deterrence, or maybe pheromones for pollination or attracting a mate, or potentially just as antibiotics to reduce infestation. So when you combine all these with the environmental factors that inevitably exist in nature, these kind of complexities really aren't suitable for, traditional FDA drug development. And one of the main limitations, I would say, of natural products is essentially the scalability. So one of the drugs, Taxol, a microtubule stabilizing agent that was actually developed partly at my alma mater at Florida State, this compound is produced by the yew tree in very low quantities and only produced in the bark of the tree itself. And by harvesting the bark, you actually kill the tree, and they're very slow growing. So obviously the true natural production wasn't going to work. So in the early days, they developed semi-synthetic methodologies in order to produce these compounds at scale. I mentioned some of the reproducibility issues. When you have a biological system, no matter how controlled you think you have it, there's always going to be some variability. So this is a limitation, along with the stability of the compound. So many of these natural products are produced in situ or on demand in a cellular environment that does not really replicate the way these compounds are produced for a drug purpose sitting on a shelf. So when you combine all these factors, really natural products serve as a good starting point, but obviously some optimization needs to happen. So going through the past 100 years of some FDA approved compounds that are naturally derived, we'll start here on the top left at the cannabinoid class. So dronabinol is synthetically produced THC, actually a racemic version of THC. Nabilone is a synthetic derivative of THC, and CBD was recently approved for epilepsy. Next, you have the indole alkaloids. The triptans are fairly well known anti-migraine and headache medications. Psilocybin and psilocin are the active ingredient from magic mushrooms. And we'll talk just in a minute about some of these compounds, including the antidepressants. You've got the ergoline alkaloids. These are generally derived from the ergot fungus produced in wheat, and these really served as a starting point for the development of LSD. Some of the other alkaloids here to the right, morphine, codeine, hydrocodone, these are all analgesics. Methadone, obviously, looking to reduce some of the addictive properties of some of these compounds. And scopolamine and atropine are used for anti-nausea medications, but are now being looked at for other CNS-type disorders. The phenethylamines on the bottom left are a very interesting drug class, and pseudoephedrine is a nasal decongestant. Albuterol is an airway bronchodilator. You've got Adderall, another CNS agent, MDMA, a psychedelic compound. These isoxazolines, isoxazoles, are actually derivatives of muscimol, which is derived from the Amanita muscara fungus. You've got conotoxin, which is a toxin from the cone snail, but is actually looked at for analgesic purposes in lieu of the opioids. And ketamine would be one of the non-natural examples that we'll get to here in a minute. So looking at mental health, it is a pretty wide-ranging, encompassing class of indications, and kind of broke this down into two main categories in which we're focusing. The first of which is mood disorders, and depression now affects 16.1 million adults, and this is nearly 7% of the US population. Anxiety disorders affect another 7 million adults within the US. And what you see with these mood disorders, and I'm also going to include post-traumatic stress disorder in here as well, is it's very unlikely that you're going to have just one mental health illness. Commonly, you'll have more than one, and substance abuse is one of these comorbidities with mental health as a coping mechanism. And what we've seen last year with the COVID-19 pandemic, we've also seen the emergence of a mental health pandemic. And just last year, the CDC reported that 40% of adults have struggled with mental health or substance abuse. And even more staggering, 1 in 10 adults have contemplated suicide just last year. Another mental health disorder that we're focused on at Psilera is cognitive disorders. Now Alzheimer's disease affects over 5 million adults within the US, and it is the 6th leading cause of death in the US, and is by far the fastest growing cause of death in the US. And as we start to develop better treatments for cardiac illnesses and other things such as cancer, as people live longer, things like these neurodegenerative disorders will become more prevalent. And unfortunately, with a lot of these mental health disorders, there aren't great treatments in order to modify the disease state. And, as these mental health disorders continue to grow, so does the market opportunity. So I wanted to outline the timeline of the antidepressant developments. And initially in the late 1950s, monoamine oxidase inhibitors were developed. Not long after, you had the tricyclic antidepressants. And then you had a couple of decade lull until you had the serotonin reuptake inhibitors in the early 1980s. And this really was kind of revolutionary at the time, and really trying to chemically modify the serotonin system and, there was certainly some efficacy, but really this timeline stops in, the early 1990s, and we haven't had any new innovations in this area for, close to 4 decades now. And one of the reasons I bring this timeline up is, so LSD was first synthesized in 1938. However, it was being used in the 1960s and 70s as an adjunct during psychotherapy in order to really get to the root cause of some of the psychiatric conditions, depression, anxiety, traumas, and things like that. However, with the passage of the Controlled Substances Act within the US, this effectively banned the research of many psychedelic compounds up until very, very recently. So the psychedelic drug class, and to meet the definition, really is just a 5-HT2A agonist. And this is the main serotonin receptor subtype that is responsible for the psychedelic effects of a lot of these compounds. And when you look at the chemical structure of serotonin compared to some of the psychedelics, which I've highlighted, obviously they're very structurally similar. You've got the indole base of a lot of these compounds. So it isn't just strictly a 5-HT2A agonism effect. You have a lot of the different serotonin subtypes, 14 of them now, and all of these compounds have varying levels of effect on these other subtypes. So you do see some polypharmacology here. And really what we have now that we didn't have back in the 1960s and 70s is a way to really identify how these compounds work within the brain. And recent studies have now really indicated a novel mechanism of action in an area where we desperately need innovation. And these compounds work through essentially a neuroplastic effect and the ability for your brain to adapt and change. And for adults, this is actually a fairly uncommon occurrence. So the default mode network is generally something that every person ruminates internally as they're not actively engaged in a task or in a conversation. However, when you have depressed or people with trauma, generally this default mode network is quite negative. And what psychedelics have the ability to do is actually to disrupt the default mode network and create a sort of the neuroplastic change. So if you look down at the bottom left, it's a very well-known graphic from Johns Hopkins University of fMRI scans of patients undergoing either placebo or psilocybin treatment. So when you're looking at the dots, these indicate different regions of the brain, and the lines connecting them would be essentially the communication between the various brain regions. So you see a lot of new pathways being developed and new communication tools within the brain, and this actually creates new neural pathways that weren't already there. So along with the personalized psychotherapy aspect of psychedelic treatments, you also have these chemical and physical changes that are happening within the brain. And this creates a very exciting opportunity to develop new therapeutics. So looking at some of these psychedelics now that have been either approved or in late stage clinical trials, ketamine is well known as an anesthetic. It's an NMDA antagonist. The S enantiomer of this ketamine was actually approved in 2019 for treatment-resistant depression. And this is clinically administered as a nasal spray, and patients undergo, close observation by the clinicians during the course of this study. And this compound is projected to reach blockbuster potential in the next couple of years. However, you're seeing a rapid uptick in ketamine clinics, and these would be intravenous racemic infusions of ketamine, essentially a generic use of esketamine, which probably will hamper the sales. But also, I think some of the newer developments of some of these psychedelics, such as MDMA, are likely to probably eat into some of these sales. So MDMA, commonly known as ecstasy, is an indirect serotonin and dopamine agonist. It's currently in phase 3 trials for PTSD, and this is being actually ushered by a nonprofit called MAPS. And in their phase 2 studies, they showed that 67% of participants did not meet PTSD criteria, and then their long-term follow-up was about a year after their initial dosing regimen. And this regimen, similar to ketamine, is administered inpatient, and about 2 to 3 doses of MDMA are useful for about a year's time looking at some of these long-term endpoints. Similarly, psilocybin is in phase 2B trials for treatment-resistant depression. You see a similar mode of action with this compound as well. You show a reduction of depression scores for up to 6 months after 1 or 2 doses. And really what you see with both MDMA and psilocybin is a very prolonged duration comparatively to ketamine. Ketamine in its state for depression is commonly administered weekly to biweekly in that type of regimen, where MDMA and psilocybin seems to be only 1 or 2 doses are needed per year. And sort of these new discoveries and some of this clinical relevance have really ushered in a new era of drug development using these compounds. And also psilocybin has been investigated for substance abuse, as well as end-of-life care, anxiety, and other palliative conditions. So at Psilera Bioscience, I'd like to say we're ushering a new era of mindful medicine. And Psilera was created both by myself and longtime colleague, Dr. Jackie Salm. We're both two PhD natural products chemists. And together we have over 20 years of experience in drug discovery as well as drug formulation. Before this, we had a career in pharmaceuticals as well as the cannabis industry. And throughout our time, we've developed a wide variety of patents. And with our last job in cannabis, created a patented product line with over $100 million in revenue in the last few years. Our company is housed within the University of South Florida's USF Connect incubator, and we've recently gathered approval to research Schedule I substances such as DMT, psilocybin, and psilocin. So this is a pretty landmark discovery or milestone within the U.S., as these compounds are Schedule I substances and hard to get a hold of. So, we are based actually in Tampa, Florida, so we like to do all of our development as well as our clinical development within the U.S., and a lot of others have done this abroad, just because the drug accessibility is much more favorable in some of these other jurisdictions. And we have a diversified drug pipeline. We're developing new chemical entities and using computational chemistry in order to aid some of these efforts. As well, we are repurposing known psychedelics using new formulation techniques. And we have now gathered a team of very powerful partners. So looking here at the right-hand side at our new chemical entities and our brain, which is our computational database. So a lot of our early work being done now is at the University of South Florida. We have people within the Neuroscience Institute as well as the Alzheimer's Institute in order to help analyze and screen some of these compounds. And we also have a biomolecular physicist that's helping us develop our computational platform. Once we do these early screening studies at USF, we are entering into a research agreement with the National Institute of Drug Abuse. And once we identify a lead compound from our screening, we'll be giving this compound to NIDA for further characterization to get us into an IND-enabling studies. And then on the left-hand side, we're also developing a new DMT delivery device. And we've partnered with the University of Wisconsin-Madison. The team up there is quite versed in psychedelic psychotherapy. They've worked with psilocybin as well as MDMA. And we've already identified partners on the CRO side, as well as a drug manufacturer in order to really facilitate these phase 1B trials. So looking at our NCEs, so we have identified the tryptamine as the scaffold of choice just because it has a very good safety profile and it mimics serotonin itself. So we will be doing some receptor screening. This would be experimental, and we're also developing this computational database, as I mentioned. So we will be taking our experimental data and comparing it and developing our computational platform in order to validate what we're seeing from our molecular modeling. Once we get through this early screening and find a compound with the target product profile in which we like, we'll be doing a bunch of animal screening, dose-finding studies. Initially, we'll be looking to reduce alcoholism in mice. And again, once we identify a lead compound, we'll be going with NIDA in order to facilitate an IND submission. So we're developing our computational platform in which we call BRAIN, the Bioreceptor Activity Intelligence Network. And with this, what we're doing is we're connecting the serotonin activity with the drug properties, chemical, physical properties, and ultimately leading into chemical space so that we can develop new psychedelic analogs and optimized compounds that can be targeted for specific indications. So we'll be using receptor binding at serotonin and some solubility and off-target toxicity effects through our computational screening. This will provide us with early validation and more of an idea of how these will work in vivo, as well as the clinical potential. And computational chemistry obviously has its advantages in terms of throughput. And as we continue to develop this database and generate more data, it will actually become more predictive and more powerful as we continue to optimize our drug development pipeline. So here is some data from our initial brain screening. We've only really labeled some known psychedelics here, but I want to start you on the graph at the very lower left of the screen. And this is the 5-HT2A system. And there is a crystal structure of 5-HT2A with LSD bound to it. So this is what we are using as our model. And the blue line is the experimental data of various psychedelics, LSD, psilocin DMT, 4-methoxy-DMT, and LSD. And what we see is actually a very good correlation from experimental Ki values that were developed cellularly to what we're seeing from our molecular modeling. And just to the right of that, we have some 5-HT2A binding scores. So in green, we have these are known psychedelics that are 5-HT2A agonists. And in blue, these are our new chemical entities in which have a very similar binding profile to the known psychedelics. So largely, these would be similarly acting compounds with psychedelic effects. However, the orange bars indicate compounds which have a reduced 5-HT2A binding, which can lead to a lower psychedelic effects. And really what we're trying to do with this type of screening is identify compounds which we can dose as outpatient therapies. And we believe this will increase access to patients that need them while combining the therapeutic effects of some of these compounds. And up at the top right, we've done some ADME studies looking at various known psychedelics at this point, as what we're displaying, of compounds that have blood-brain barrier permeability. Obviously, we're targeting CNS disorders, so we need compounds to get into the brain. And in white, we have intestinal permeation. So these are some other known psychedelics. These are generally charged compounds, so can pass through the intestinal membrane but not get into the blood-brain barrier. As well as we can start to identify off-target effects such as P-glycoprotein effects and start to really screen and optimize our R&D efforts and synthetic efforts to get these into preclinical and clinical trials. So one of the ways, well, there are a myriad of ways in which you can develop new drugs, part of which is starting with natural products isolation. We've talked a little bit about SAR and different ways that you can modify compounds in order to optimize your drug itself. Another way that we're looking to tackle the problem of psychedelics as they exist today is with optimized dosing regimens. So Epidiolex, right in the center of the screen, is actually cannabidiol derived from cannabis. And this compound essentially has no water solubility. It's an oil-based formulation, low permeability, would be defined as a class 4 compound. So not one that's great for drug formulators, hence why it has a very low bioavailability. But Dr. Salm and I, from our previous experience in cannabis, we developed new and innovative ways in order to deliver compounds like CBD and THC. And this would be through novel inhalation techniques such as meter dose inhalers and nebulizers, as well as transdermal delivery devices. And these would bypass a lot of the metabolism, as well as the absorption effects that happen with these compounds. Similarly, migranol is an ergotamine derivative, and this compound is dosed intranasally. And this is done because intranasal is nearly a direct blood-brain barrier delivery device. So this is another way that you can improve bioavailability. And the last thing I'll note here is we mentioned psilocybin, the active ingredient from magic mushrooms. However, this compound, the natural product, is not actually the active ingredient within the system itself. Once you ingest psilocybin, it is rapidly dephosphorylated into psilocin, and this is actually the active compound that induces the antidepressive effects. So, when you're looking at this sort of pro-drug approach, there's a lot of ways that you can optimize some of these compounds. So why DMT? And this is one that we are definitely focused on at Psilera. It is the only endogenous psychedelic compound. It is produced between a lot of mammals as well as humans, and throughout nature and plants and other organisms. It's a very simple compound. It's cost efficient to produce, and it's the only psychedelic to increase neuronal growth at sub-psychedelic dosages. And this is very important as we're trying to develop outpatient therapies. You look at the right of the screen at some of the rat neurons treated with either DMT or the vehicle. You see a lot more branching as well as neuronal growth with the cellular types. And as well, in vivo, these compounds show improved memory and learning in animals. So one of the major issues with DMT, I would say, is its lack of bioavailability. So this compound actually can't be dosed alone orally because it is rapidly metabolized by MAOs in your body. So one of the ways that it's commonly done, I would say holistically, is through ayahuasca, which contains MAOIs, so this compound can be dosed orally. However, once you start combining drugs, obviously from an FDA perspective, it becomes a little more challenging in order to get these therapies approved. So many people today are undertaking clinical development of DMT using an intravenous dosing method. However, with our backgrounds in formulation, we're looking at non-invasive ways to deliver DMT either through very short-acting delivery devices, whether they be to induce the psychedelic effects. And because DMT has a relatively short half-life, these would be a quick onset as well as a quick offset in terms of duration. And from a clinical perspective, this is very important. Additionally, we're developing another innovative delivery system in which we can produce more of a sustained multiple day therapy. And this would be below the threshold to induce psychedelic effects, but still there in order to have the therapeutic potential of DMT itself. And we believe when you combine these aspects that this drug can be very useful for things like mood disorders. Most psychedelics have mood calming effects due to their activation of the serotonin system, as well as increasing neuronal growth for things like dementia, Alzheimer's disease, and increasing some of the memory and cognition issues there. So we're really excited about this new innovative delivery system, and we are in the process of submitting an IND as well as begin recruiting patients by the end of this year for a phase 1b clinical trial. So I'll wrap up here by saying Psilera's slogan: we are reimagining psychoactive natural products as building blocks for the next generation of neurological, CNS, and psychiatric disorders. It's time to bridge the 21st century drug development with over hundreds and frankly thousands of years of psychedelic research. I want to thank you for your time, and I'm happy to answer any questions. Thank you.
Great presentation again, Chris, and overall very good overview of the CNS situation, like home here at the USA and globally abroad. And I will open up the floor now to any questions for Chris. You can use the Q&A and discuss tab to the right. Also, Dr. Duss, if you have any remarks, feel free to add them as well.
Hey, Chris, great talk. I really like the fact that you presented about the crystal structure of the 5-HT receptor. That's kind of work we do that I'm going to talk about a lot tomorrow, and target engagement of molecules with crystal structures and so on. So it was nice to see it as part of your talk. What's next? Are you looking for funding?
Yeah, we are actually in the process of closing our funding round as we speak. There is a lot of interest within the space, and we've identified some really, really great investors that see our vision. And, really this is just kind of the early stages of where psychedelics could be down the road. And obviously with mental health the way it is, there's not going to be any lack of opportunities. And these compounds, like I mentioned, they have a novel mechanism of action for a lot of things that need innovation. So we're really excited to kick off this journey to begin some of these clinical developments. We've done a lot of preclinical R&D work. Fortunately, I can't share a lot of it due to patentability issues, but we're really, really excited to be here and to present some of this research.
Great, thanks a lot.
Amazing. Thank you again, Chris. And you can find more information about his team on LinkedIn. You can also email us, and we can share details about his group. So thank you again, Chris, for your presentation. And we're just going to head over now, guys, to a quick break. So what we have right now in our agenda is a quick 20-minute break until 2:05 p.m. Pacific time. which is about 5:05 p.m. Eastern Time. And then we'll continue the afternoon with 4 more presentations, Prem from Benchling, Dr. Woody Sherman, CSO of Silicon Therapeutics. He was recently in the news because his group was, I believe, acquired by Rovent Sciences. Then we have Shruti from Novartis, and then Sanja, Dr. Sanja from Sana Biotech will be our final keynote speaker. Then we'll have our panel discussion from 4:05 p.m. to 5:30 p.m. Pacific Standard Time. Also, during this networking break, if you want to take this opportunity to interact with other users, you can do so by visiting the lobby. If you see my screen, you can message people over here, share your business card, etc. You can go to the lounge area as well. You can go to the booths. Most importantly, visit a couple booths that you're interested in, and overall take advantage of this one-on-one networking session. A special thanks again to our sponsors for their support. And with that said, we will be heading for a quick break until 2:05 Pacific Time.